Before you think about forests, or oceans, or endangered species moving like shadows through tall grass, you begin somewhere much smaller. You begin inside yourself. There is a quiet in your body that you rarely notice. Beneath your breathing, beneath your heartbeat, beneath even your thoughts, there is another rhythm. It is slower than a blink and older than memory. It lives inside nearly every one of your cells, curled into a delicate spiral known as DNA. If you could shrink down smaller than a grain of sand, smaller than a speck of dust floating in evening light, you would find it there. A long, coiled molecule folded carefully so it fits inside a space far tinier than your eye could ever see. And yet this slender thread carries instructions for building you. Not just your height or the color of your eyes, but the quiet architecture of your skin, the rhythm of your blood, the shape of your smile. DNA is often described as a code, but tonight you can imagine it as a language. It is written in 4 chemical letters, A, T, C, and G: adenine, thymine, cytosine, guanine. The names sound complex, but the idea is simple. Like letters in a book, these 4 symbols repeat in different orders, forming words. Those words form sentences. And those sentences form chapters, not of stories printed on paper, but of living tissue and beating hearts. Inside your cells, this language is always being read. When a cell needs to build something, perhaps a protein to repair a tiny tear, or an enzyme to help digest your food, it does not guess. It does not invent. It reads the instructions stored in DNA. A small molecular machine copies a section of the code into a messenger molecule called RNA, like taking a handwritten recipe from a heavy cookbook so it can be carried into the kitchen. Then other tiny structures, called ribosomes, follow the recipe step by step, linking together amino acids into proteins. Proteins are the quiet workers of life. They shape your muscles. They carry oxygen in your blood. They help your neurons send signals that become thoughts. Every movement you make, every breath you take, is guided by instructions written in that 4-letter alphabet. And yet, the same alphabet writes forests. The DNA in a tiger's cells follows the same chemical rules as yours. The DNA in a coral reef, glowing softly beneath ocean waves, is written in the same 4 letters. Life, in all its forms, shares this common script. The differences between you and a whale, between a pine tree and a butterfly, are not in the language itself, but in the arrangement of its letters. You can picture it like music. If the 4 letters are notes, then each species is a different melody. Some melodies are fast and bright. Others are slow and deep. But all are composed from the same small set of tones. Within this melody are units called genes. A gene is a segment of DNA that contains instructions for building a specific protein. You might imagine a gene as a paragraph within a much larger book. Some paragraphs describe how to form fur. Others describe how to detect light. Others still determine how a body responds to cold or heat, to hunger or illness. Genes do not float in isolation. They interact. They turn one another on and off, like lights in a vast city seen from above. In early development, certain genes activate in careful sequences, guiding a single fertilized cell to divide, to specialize, to become bone, skin, brain, and blood. It is not chaos. It is choreography. And yet, it is not rigid either. Within the DNA of every species, small variations exist. Tiny differences in the order of letters can change how a protein folds, how efficiently it works, how strongly it responds. These variations are called mutations, though the word need not feel alarming. Most mutations are neutral. Some are harmful. A few are helpful. Over long stretches of time, helpful variations accumulate, shaped gently by natural selection, the process by which traits that support survival become more common. You can think of natural selection as a quiet editor. It does not write new sentences from nothing. Instead, it reviews countless drafts generated by variation and preserves the ones that help a species endure. Over generations, this editing shapes wings from forelimbs, lungs from simple sacs, eyes from light-sensitive cells. But the editing takes time. Vast, patient time. Tonight, as you rest, consider how every endangered species also carries this language within its cells. A snow leopard padding silently across icy ridges holds DNA shaped by thousands of years of mountain wind. A rare orchid blooming in a shrinking rainforest carries genetic instructions refined through ages of sunlight and soil. Conservation, then, is not only about landscapes or numbers. It is about preserving these living libraries of information. When a species disappears, an entire melody falls silent. A unique arrangement of A, T, C, and G never to be written again in quite the same way. Inside each organism is a record of adaptation, of resilience, of survival against odds you can scarcely imagine. And yet, the language itself is fragile. DNA must be copied each time a cell divides. Most of the time, this copying is astonishingly accurate. Specialized enzymes proofread the sequence, correcting mistakes. But sometimes, an error slips through. A letter changes. A small deletion occurs. Usually, it does not matter. Sometimes, it does. In small populations, where few individuals remain, harmful variations can accumulate more easily. There are fewer alternative versions of genes to balance them. The melody grows narrower, less varied. The resilience that once allowed adaptation to changing climates or emerging diseases can thin. Still, at its heart, DNA remains a message. It is both ancient and immediate. You carry sequences inherited from ancestors stretching back beyond recorded history, beyond the invention of language, beyond even the first mammals. And yet, in this very moment, your cells are reading that code anew, translating it into living action. There is something deeply humbling in this. You are not separate from the natural world. You are written in the same script as coral reefs and redwood trees. The same chemical letters that form your heartbeat also shape the flight of birds and the shimmer of fish scales beneath moonlit water. So when you think about saving endangered species, you are not imagining something distant. You are thinking about protecting variations of a language you already speak at the cellular level. Tonight, you simply listen. You imagine the double helix, two strands entwined like a soft spiral staircase, turning quietly inside every living thing. You imagine genes switching on and off like lanterns in a vast, dark landscape. You imagine proteins assembling with patient precision, sustaining life moment by moment. And you begin to sense that conservation does not begin in forests or oceans. It begins here, in the cell. Because if life is written in a language, then to protect life is to protect its words. To understand the script. To notice where letters have faded. To recognize when entire sentences risk being erased. In the chapters ahead, you will step outward from the cell into habitats, from DNA into ecosystems, from molecules into mountains. But for now, you rest with the smallest scale. With the quiet alphabet. With the spiral thread carrying instructions older than mountains. And as you drift gently towards sleep, a question begins to form: what happens when those living libraries grow smaller, when pages are lost, when entire species stand at the fragile edge of survival, holding only a fading fragment of their once vast genetic song? You step outward now. Beyond the quiet spiral inside a single cell, beyond the soft alphabet of A, T, C, and G, you lift your gaze to landscapes. To coastlines that curve like sleeping animals. To forests that breathe in long green sighs. To grasslands where wind moves in silver waves. And yet, beneath this beauty, there is a thinning. Not always visible at first. Not always loud. Extinction rarely announces itself with thunder. More often, it arrives like silence spreading slowly across a room. You walk, in your mind, through a rainforest. The canopy is still tall, still shimmering with filtered light. But fewer wings flutter between the branches. Fewer calls echo at dusk. The forest stands, but some of its melodies have softened. Populations shrink in ways that are difficult to see. A species that once counted in hundreds of thousands may now number in mere hundreds. A coral reef once vibrant with branching color may bleach pale under warming seas. A herd that once stretched to the horizon may now fit within a single valley. And when numbers fall, something else begins to fade. You remember that DNA is a language. Within every species, that language contains variation, small differences in the order of its letters. These variations are not flaws. They are possibilities. One gene variant might help tolerate cold. Another might offer resistance to a particular disease. Another might influence fertility or strength or metabolism. Genetic diversity is like a vast library. Each individual carries a slightly different edition of the same book. When populations are large, the library is full. Many versions exist. Many possibilities rest on the shelves. If the climate shifts or a new pathogen appears, there is a greater chance that somewhere in that library, a useful passage exists, a sequence of DNA that allows survival. But when populations shrink, the library loses books. Imagine a species reduced to just a few dozen individuals. The range of genetic variation narrows. Certain gene versions disappear entirely, simply because the individuals carrying them did not survive or reproduce. The shelves grow emptier. The story becomes less varied. This is not just poetic imagery. It is population genetics, the study of how gene frequencies change in groups over time. In small populations, a process called genetic drift becomes powerful. Drift is random change. It is not guided by survival advantage, but by chance. If only a few individuals reproduce, their particular versions of genes become more common, even if those versions are not especially beneficial. Other versions may vanish purely by accident. Over generations, this randomness can reduce diversity further. You might think of it as shuffling a deck of cards again and again, but removing several cards each time. Eventually, the deck contains fewer possibilities. Certain combinations can no longer be dealt. And with fewer combinations, adaptability declines. You imagine a species of frog living in cool mountain streams. For centuries, its genes have quietly supported life in stable conditions. But then a fungal disease arrives, spreading through moist skin. If within the population there are gene variants that confer resistance, perhaps subtle changes in immune response, those frogs are more likely to survive and reproduce. The population can recover. But if diversity has already narrowed, if those protective variants were lost in earlier declines, the species may have little defense. The disease moves through like wind through dry leaves. This vulnerability is called inbreeding depression. When close relatives reproduce, harmful recessive mutations, usually hidden, can pair up and express themselves. Fertility may drop. Lifespan may shorten. Offspring may struggle to survive. The melody of life grows softer, less resilient. And still, this happens quietly. There are no alarms in the forest. No flashing lights across the savanna. Just fewer nests each spring. Fewer tracks in the snow. Fewer seeds carried by wind. You consider how human activity shapes this thinning. Habitat fragmentation divides continuous landscapes into isolated patches. Roads, farms, cities, they create barriers. A once connected population becomes several small ones, each with its own shrinking library of genes. Isolation matters. When individuals cannot move between groups, gene flow, the exchange of genetic material, slows or stops. Variation that might have spread across a species remains trapped in one small pocket. If that pocket disappears, its unique genetic pages vanish with it. And climate change adds another layer. As temperatures rise and weather patterns shift, species must adapt, migrate, or decline. Adaptation depends on variation. Without sufficient diversity, even subtle environmental changes can overwhelm. You begin to see conservation differently. It is not only about protecting individual animals. It is about protecting variation itself. Protecting the breadth of the library. Maintaining enough copies, enough differences, that life retains options. Scientists measure this diversity using genetic markers, small, identifiable sequences within DNA. By comparing these markers across individuals, they estimate how much variation remains. They can see bottlenecks, moments in history when populations dropped sharply and diversity narrowed. A bottleneck is like a river forced through a thin canyon. Much of what once flowed freely is compressed. On the other side, the river may widen again, but some of its earlier breadth is gone. Many endangered species today carry the signature of such bottlenecks. The northern elephant seal, hunted nearly to extinction in the 19th century, now numbers in the hundreds of thousands again. Yet genetically, the population is remarkably uniform. The library rebuilt in size, but not in variety. Size alone is not enough. As you rest with this, you feel both the fragility and the quiet persistence of life. Even in reduced numbers, species continue. They breed. They forage. They navigate landscapes altered beyond recognition. There is dignity in that endurance. But endurance without diversity can be precarious. You return, briefly, to the cell, to DNA, to the letters repeating in careful sequence. In a large, healthy population, those sequences differ subtly from individual to individual. In a diminished one, the difference is narrow. And so the question emerges gently. If a library has lost books, can it be replenished? If variation has faded, can it be restored? Can science, which now reads the language of DNA with increasing clarity, also help rewrite or reintroduce missing pages? This is not a question of domination. It is a question of possibility. For decades, conservation has focused on habitat protection, breeding programs, anti-poaching efforts, all vital, all essential. But now, as genetic tools grow more precise, another approach begins to shimmer at the horizon. What if, instead of only preserving what remains, we could carefully strengthen it? What if the language inside the cell, the same language you carry within your own body, could be gently edited to reintroduce resilience? You do not rush toward an answer. You simply hold the question. You stand at the fragile edge of survival with these species, feeling both the weight of loss and the spark of curiosity. The libraries are thinning. The melodies are quieter. And somewhere, in a laboratory not far from forest or reef, scientists are studying a molecular system first discovered in bacteria, a system that remembers invaders and cuts DNA with astonishing precision. A system that may allow humanity, for the first time, to edit the script itself. And as you breathe slowly, letting this thought settle, you sense that the next step in this journey will take you not deeper into wilderness, but deeper into the cell once more, where a molecular pair of scissors waits in patient silence. You return now to the smallest scale. Back to the cell. Back to the spiral staircase of DNA. Back to the quiet language written in 4 steady letters. But this time, you are not alone in that microscopic world. You are standing beside bacteria, ancient, single-celled organisms that have lived on Earth for billions of years. Long before forests rose. Long before animals walked. Long before humans began to wonder about genes, these tiny beings were surviving in oceans, soil, and even within your own body. Bacteria live in a world of constant threat. Viruses, far smaller than they are, drift through water and air, seeking entry. When a virus infects a bacterium, it injects its own genetic material, hijacking the cell's machinery to make copies of itself. It is an invasion swift and ruthless. And yet, bacteria are not defenseless. Within their DNA, scientists discovered peculiar repeating sequences. Short palindromic patterns, segments that read the same forward and backward, spaced regularly along the genome. At first, these repeats seemed mysterious. They were named CRISPR, an acronym for clustered regularly interspaced short palindromic repeats. The name is technical. But the idea is gentle. Between those repeating sequences lie fragments of viral DNA, tiny genetic souvenirs from past infections. When a bacterium survives a viral attack, it sometimes captures a snippet of the invader's DNA and inserts it into its own genome, nestled between the repeats. It is as if the bacterium keeps a scrapbook. Each viral fragment is a pressed flower, carefully stored. A memory of an enemy once encountered. If the same virus attacks again, The bacterium transcribes that stored fragment into RNA, a messenger molecule that carries the memory outward. This RNA then partners with a special protein called Cas. One of the most well-known is Cas9. You can imagine Cas9 as a pair of molecular scissors. But not wild or reckless scissors. Not blades swinging freely. Instead, they are guided with extraordinary precision. The RNA acts like a compass or a guidebook, leading Cas9 to a matching sequence within the invading viral DNA. When the match is found, Cas9 cuts. The viral genome is sliced apart. The invasion is halted. The bacterium survives. For countless generations, this quiet defense system operates unnoticed by human eyes. A microscopic immune system, elegant and efficient, protecting simple cells in complex environments. Then, in the early 21st century, scientists begin to understand its potential. If bacteria can use RNA to guide a cutting protein to a specific DNA sequence, could humans do the same? Could researchers design a custom RNA guide, one that matches a gene of their choosing, and direct Cas9 to cut at that precise location? The realization feels almost still, almost suspended. Because cutting DNA is not a trivial act. DNA is the instruction manual of life. To cut it is to intervene in the script itself. Yet cells are not fragile manuscripts that crumble at the slightest tear. They possess repair systems. When DNA is cut, cellular machinery rushes to mend the break. Sometimes, the repair process introduces small changes, adding or deleting letters. Sometimes, if scientists provide a template, the cell can incorporate new sequences into the cut site. In this way, CRISPR-Cas9 becomes more than bacterial memory. It becomes a gene editing tool. You picture it clearly. A designed RNA molecule, carrying a sequence chosen by researchers, floats within a cell. It binds to Cas9, forming a complex guide and scissors together. The RNA searches along the vast stretches of DNA, scanning for its matching letters. When it finds the correct sequence, Cas9 makes a precise cut. Not everywhere. Not randomly. But at a specific, intended site. The simplicity is part of its power. Earlier gene editing techniques existed, but they were often complex and costly. CRISPR is comparatively straightforward. By changing the guide RNA sequence, scientists can retarget Cas9 to different genes with relative ease. It is modular, adaptable. You feel the weight of that. To edit DNA is to adjust the instructions that shape living organisms. In medicine, this has opened possibilities for correcting genetic disorders in cells. In agriculture, it has allowed the development of crops with improved resilience. And in conservation, the realm you are slowly approaching, it suggests something profound. If a species is vulnerable because of a specific genetic weakness, could CRISPR help repair it? If harmful mutations have accumulated in a small population, could targeted edits restore function? If disease threatens to erase a lineage, could resilience be reintroduced at the molecular level? But before those questions fully unfold, you linger in the science. CRISPR does not rewrite entire genomes wholesale. It works at defining points. Precision matters. Scientists must ensure that the guide RNA matches only the intended sequence, avoiding off-target cuts elsewhere. Research continues to refine accuracy, to develop new variants of Cas proteins that cut more cleanly or even modify DNA without making full breaks. There are versions now that act like pencils instead of scissors, base editors that change a single letter without slicing the double helix. Others insert sequences more gently. The technology evolves rapidly, but its foundation remains that bacterial memory system, a defense mechanism repurposed. You imagine the first moment a researcher watches CRISPR successfully edited gene in a laboratory cell. Perhaps there is quiet awe. Perhaps caution. Perhaps both. The realization that humanity can now adjust the letters of life's alphabet with increasing precision is not dramatic like a storm. It is quieter, more intimate. Like discovering you can revise a sentence in a book that was once thought unchangeable. And yet, revision carries responsibility. In bacteria, CRISPR evolved through natural selection, shaped over eons to serve survival. In human hands, it becomes intention-driven. We choose the target. We design the guide. We decide where to cut. You sense that this is not merely a technical achievement. It is a threshold. To read DNA was one step. To sequence entire genomes, mapping the letters, expanded understanding. But to add it? To alter the sequence itself? That shifts the relationship between humanity and biology. You are not separate from the language of life. You are fluent in it at the cellular level. And now, as a species, you are learning to write within it. You let that settle gently. Because the goal, in conservation, is not control. It is preservation. Not domination, but care. And yet, care sometimes involves intervention. A surgeon cuts to heal. A gardener prunes to strengthen growth. CRISPR, in its essence, is a molecular instrument. Neutral. Precise. Capable of harm or help depending on how it is used. In the chapters ahead, you will see how this instrument might be applied to species weakened by disease, to populations thinned by genetic bottlenecks. You will consider frogs vulnerable to fungal infections, bats threatened by pathogens spreading in dark caves. For now, you simply understand the tool. A memory system from bacteria. A guide RNA acting as compass. Cas9 serving as careful scissors. A cut. A repair. A subtle shift in the letters. And as you rest in this understanding, you feel the quiet gravity of it. Because once you realize that life's script can be edited, the question is no longer whether the scissors exist. The question becomes how and when they should touch the fragile melodies of species already struggling to endure. You sit quietly now at the edge of a mountain stream. The water is clear, sliding over stones worn smooth by centuries. Moss clings to the banks. The air is cool, almost still. If you listen closely, you can imagine the faint sound of frogs, or perhaps, the absence of it. In many places around the world, frogs have grown silent. A fungal disease called chytridiomycosis has spread across continents, carried unknowingly through global trade and travel. The fungus infects amphibian skin, disrupting its ability to regulate water and electrolytes. For creatures who breathe partly through their skin, this disruption is devastating. Entire species have declined. Some have vanished. You imagine a single frog resting on a damp rock. Inside its cells, DNA carries the instructions for its immune system, the complex network of proteins and signals that detect and fight pathogens. Tiny variations in certain genes influence how effectively immune cells recognize fungal molecules, how strongly they respond, how efficiently they clear infection. In some individuals, particular gene variants offer partial resistance. In others, they do not. This difference can be only a few letters in the DNA sequence. A small substitution. A subtle change in a protein's structure that allows it to bind a fungal component more tightly or to activate a defense pathway more quickly. A single change. A different outcome. You realize that vulnerability and resilience are not abstract qualities. They are written into molecules. Not as destiny, but as probability. Now imagine a population reduced to only a handful of individuals. If the variants that offer resistance are rare or absent, the fungus moves swiftly through them. If those protective letters once existed but were lost in a genetic bottleneck, the species faces the disease without its strongest shield. This is where the idea of gene editing enters conservation not as ambition, but as a question of repair. If scientists can identify gene variants associated with resistance, perhaps in closely related species that survive infection, could those variants be introduced into vulnerable populations? Could CRISPR guide the molecular scissors to a specific gene and adjust a sequence so that the resulting protein functions more effectively? You do not imagine dramatic transformations. No glowing creatures. No unnatural forms. Instead, you picture something subtle. A small edit in an immune-related gene. A corrected mutation that restores a protein's stability. A sequence change that mirrors one already found in nature among resistant individuals. In laboratory settings, researchers have begun exploring these possibilities. They sequence genomes from both susceptible and resistant populations, searching for differences. They test how particular gene variants influence immune responses. Slowly, patiently, they map the landscape of vulnerability. You shift your attention now to bats. In North America, white nose syndrome, caused by a fungal pathogen, has devastated bat colonies. The fungus grows on the skin of hibernating bats, disrupting their metabolism and causing them to wake frequently during winter. Their stored fat depletes. Many do not survive until spring. Some bat populations show signs of emerging resistance. Survivors may carry gene variants that help regulate immune responses or manage energy use more efficiently during infection. Again, variation matters. Again, a few letters in DNA can tilt the balance. The concept of editing these genes is not simple. It requires understanding which sequences truly influence resistance and ensuring that changes do not create unintended consequences. Genes rarely act alone. They exist within networks, interacting with others in ways that are sometimes predictable and sometimes not. You think of it like adjusting a single string on a violin. Tighten it slightly, and the pitch shifts. But the sound of the instrument depends on all its strings, on tension balanced across the frame. Altering one note affects the harmony. CRISPR provides precision, but biology provides complexity. Scientists must test edits in controlled environments. They examine whether modified cells behave as expected. They monitor for off-target effects, unintended cuts elsewhere in the genome. They assess long-term stability. They ask whether edited traits will persist across generations without harming other aspects of fitness. This is slow work. It is deliberate. Ethical review boards weigh risks. Ecologists consider ecosystem dynamics. Conservationists debate alternatives: habitat restoration, captive breeding, translocation. Gene editing is not a replacement for these efforts. It is an addition, a potential complement when traditional measures are insufficient. You rest with that balance. Imagine a captive breeding program for an endangered amphibian species. Individuals are raised in biosecure facilities to protect them from fungal exposure. Scientists identify a resistance-associated gene variant present in a related species. Using CRISPR, they introduce that variant into a small number of embryos. The resulting frogs are monitored carefully, their health and immune functions studied in detail. If the edit proves beneficial and safe, those individuals could be incorporated into breeding programs, gradually increasing the frequency of resistance in the population before reintroduction into the wild. The goal is not to redesign the species. It is to restore a capacity that once might have arisen through natural selection, but now may not have time to develop. Climate change accelerates disease spread. Globalization moves pathogens rapidly. Evolution still operates, but it may not keep pace with the speed of environmental disruption. CRISPR, in this context, becomes a way to gently assist adaptation. Yet caution remains essential. You think of ecosystems as woven fabrics. Each species threads through others: predator and prey, pollinator and plant, host and symbiont. Altering one species' genetic makeup may have ripple effects. A more disease-resistant frog might alter population dynamics of insects. A bat population rebounding could shift competition patterns. These are not reasons to avoid intervention altogether. They are reasons to proceed thoughtfully. You breathe slowly and consider the weight of intention. In the past, human influence on species often occurred without foresight: overhunting, habitat destruction, introduction of invasive organisms. Now, with tools like CRISPR, intervention becomes precise and purposeful. The difference lies not only in technology, but in awareness. You are learning to see genes not as static fate, but as dynamic sequences that can be read, understood, and in rare cases, carefully adjusted. But each adjustment is a choice. A choice to touch the script of life. A choice to intervene in evolutionary pathways shaped over millennia. As you sit beside that mountain stream, you do not rush toward certainty. You simply hold both compassion and caution. The frogs deserve survival. The bats deserve spring skies filled with insects. The ecosystems they support deserve continuity. And yet, the act of editing DNA is profound. It requires humility and acknowledgement that knowledge is always incomplete. That unintended outcomes are possible. That restraint can be as important as action. Still, the possibility lingers gently. What if resilience, once lost through shrinking populations and rapid change, can be restored at the molecular level? What if careful edits can strengthen species without reshaping their essence? You feel that the answer does not lie in a single gene or a single species. It lies in something broader, in the diversity of sequences that allow life to adapt in countless ways. Because resistance is not only about one protective variant. It is about variation itself. And as you let that thought settle into the rhythm of your breathing, you sense that the next step in this journey will move beyond single genes and into the wider constellation of differences that make populations strong, into the mathematics of diversity itself. You lift your gaze from a single gene and begin to see the sky. Not the sky above forests or oceans, but a different kind of sky, one that stretches across a genome. A vast expanse of letters, billions long, where variation glimmers like scattered stars. Each star is a difference. At a particular position in DNA, one individual may carry an A, while another carries a G. At another position, there may be a small insertion, a subtle deletion. These differences are called alleles, alternative versions of a gene or genetic sequence. Most alleles do not announce themselves loudly. They quietly influence how proteins fold, how enzymes function, how cells respond. Together, they form diversity. Genetic diversity is not about dramatic mutations. It is about accumulation. Thousands upon thousands of small variations, spread across individuals in a population, creating a reservoir of potential responses to change. You can imagine a population as a night sky. In a large, healthy population, the sky is dense with stars. Many alleles circulate. Some are common. Some are rare. But collectively, they offer flexibility. If drought comes, certain combinations may help conserve water. If a new pathogen emerges, a rare immune variant may prove protective. Now imagine that sky narrowing. When a population declines sharply through habitat loss, overhunting, or disease, the number of individuals shrinks. With them, many rare alleles disappear simply because their carriers do not survive or reproduce. The sky dims. This is more than metaphor. It is measurable. Scientists calculate something called heterozygosity, the probability that two copies of a gene in an individual are different. High heterozygosity often reflects high diversity. In small populations, heterozygosity tends to decrease over generations, especially when individuals breed with relatives. Inbreeding increases homozygosity, the presence of identical gene copies. While some genes function perfectly well in identical pairs, others carry recessive mutations that are harmless when paired with a healthy allele but harmful when doubled. Inbreeding depression emerges from this doubling. You picture it gently. A gene variant that slightly reduces fertility exists quietly in a large population, masked by healthier variants. But as diversity shrinks and relatives breed, that variant may pair with itself. Fertility declines more noticeably. Survival drops. The population weakens further. It is not fate. It is probability shifting. Mathematics, at its core, describes patterns of chance. In population genetics, equations model how allele frequencies change over time. They account for mutation, migration, selection, and drift. But tonight, you do not dwell on formulas. You feel the pattern instead. Diversity is resilience. A wide sky allows constellations to rearrange. A narrow sky limits possibilities. Now you consider the bold question: if diversity has thinned, can it be widened again? Traditionally, conservation biologists attempt to restore diversity through managed breeding programs. Individuals from different subpopulations may be brought together to increase gene flow. Captive breeding can be designed to maximize genetic variation, pairing individuals strategically to reduce inbreeding. These methods work, especially when enough diversity remains. But what if the sky has already lost many stars? This is where gene editing enters the conversation once more. CRISPR is precise at the level of individual genes. It can correct a harmful mutation. It can introduce a beneficial variant. But diversity is not one gene. It is thousands, millions of positions across the genome. You feel the scale of that. Rebuilding diversity through editing alone would not mean rewriting entire genomes. That would be neither practical nor wise. Instead, scientists might focus on key loci, genes strongly linked to fitness traits such as immune response or reproductive success. By increasing variation at these critical points, they could strengthen a population's adaptive potential without attempting to manufacture artificial complexity. There is also another possibility. If preserved DNA from historical populations exists in museum specimens or frozen tissue banks, scientists can sequence those genomes and identify alleles that have been lost. In theory, some of those lost variants could be reintroduced into living populations using gene editing. It is a delicate idea. You imagine it like restoring faded passages in an old manuscript, using fragments preserved in archives. The goal is not to create something new, but to recover something that once belonged. Still, questions arise. How many alleles are necessary for resilience? Which ones truly matter? How do edited individuals integrate into wild populations? Will natural selection maintain or remove introduced variants? These are not simple questions, and there are no universal answers. Population genetics reminds you that evolution is dynamic. Diversity is not static wealth stored in a vault. It shifts with environment. What is beneficial in one context may be neutral or harmful in another. And yet, maintaining breadth, maintaining options, increases the chance that some variants will prove useful as conditions change. You consider climate once more. As temperatures rise, precipitation patterns shift, and habitats transform, species face pressures that differ from those of the past. Adaptive potential becomes critical. Without sufficient variation, evolution has little raw material to shape. CRISPR does not replace evolution. It can, at most, provide additional material for it to act upon. There is humility in that understanding. Gene editing may widen parts of the sky, but it cannot predict every future constellation. It can introduce variation thoughtfully, but it cannot script destiny. Perhaps that is why conservation genetics increasingly blends approaches. Protect habitats. Support population growth. Facilitate natural gene flow. Use genetic rescue, introducing individuals from other populations when possible. And consider gene editing only when the need is clear and the science is sound. You return to the image of stars. In a vibrant sky, diversity feels effortless. In a diminished one, every star matters more. You breathe slowly and reflect on how interconnected this all is. A single gene can influence disease resistance. A collection of alleles across the genome shapes fertility, longevity, adaptability. Populations are not just numbers of individuals, they are networks of variation. And beneath it all lies that quiet alphabet. A, T, C, G. Rearranged across millions of positions, forming subtle differences that accumulate into resilience. As you drift toward rest, you sense that restoring diversity may require more than editing the present. You step into a room that feels almost like winter. Not the wild winter of mountains and wind, but a controlled, deliberate cold. Stainless steel tanks stand tall and still. Soft vapor spills over their rims, drifting downward like low clouds. Inside these vessels, immersed in liquid nitrogen at temperatures colder than the Antarctic night, cells sleep. This is sometimes called a frozen zoo. Within these tanks are tiny vials containing sperm, eggs, embryos, skin cells, genetic material collected from endangered species across decades. Rhinos, big cats, rare antelope, amphibians whose calls are no longer heard in their native streams. Each vial holds more than cells. It holds sequences of DNA that once moved through living bodies under open skies. Cold does something remarkable to biology. At extremely low temperatures, chemical reactions slow almost to stillness. Cellular processes pause. Time, in a sense, stretches out. Properly preserved, DNA can remain intact for years, even decades. You imagine these cells as seeds in a vault. Not seeds of plants, but seeds of variation. Within them are alleles that may no longer be present in living populations. Gene variants carried by individuals who died before breeding. Subtle differences that slipped from the sky of diversity when numbers dwindled. In earlier chapters, you felt the thinning of that sky. Here, in the frozen quiet, you encounter fragments of what was once brighter. Scientists collect and preserve genetic material for many reasons. In captive breeding programs, frozen sperm can be used for artificial insemination, allowing gene flow between individuals separated by geography or time. Embryos can be stored until suitable surrogate mothers are available. Skin cells can be reprogrammed into induced pluripotent stem cells, cells capable of developing into many tissue types. The goal is preservation of options. But preservation alone does not guarantee revival. Some species decline so rapidly that living populations are too small to recover naturally, even with stored material. Others persist, but with severely reduced genetic diversity. This is where the frozen zoo intersects with CRISPR. You imagine a scenario gently. A population of endangered wild cats has lost much of its historical genetic variation. Researchers examine preserved samples from decades earlier and sequence their genomes. They identify alleles that were once common but are now absent, variants linked to immune strength or fertility. Using gene editing, scientists could, in theory, reintroduce specific lost variants into living individuals. Skin cells from a current animal might be cultured in a laboratory. CRISPR could adjust targeted sequences to match those found in the archived genome. The edited cells could then be used to create embryos through cloning techniques or advanced reproductive technologies. It sounds complex because it is. Somatic cell nuclear transfer, the process used to clone animals like Dolly the sheep, involves transferring the nucleus of a body cell into an egg cell whose own nucleus has been removed. The resulting embryo carries the DNA of the donor cell. If that donor cell has been carefully edited, the embryo will carry the revised sequence. You sit with the precision of it. A letter altered. A gene restored. an embryo formed. Yet the aim is not to create novelty. It is to recover what once existed within the species' own evolutionary history. There are already early examples of this intersection. In some conservation projects, preserved cells from individuals long deceased have been used to increase genetic diversity in small populations. The technology is still developing, and success varies, but the principle holds: frozen genes are not merely artifacts. They are potential participants in future recovery. You think of it like tending an orchard. If a rare tree variety nearly vanished but seeds were stored decades ago, replanting those seeds would restore lost branches of diversity. Gene editing adds another layer, allowing gardeners to repair damage in the seeds themselves before planting, correcting mutations that accumulated in surviving trees. Still, care is essential. DNA does not operate in isolation from environment. Genes interact with epigenetic marks, chemical modifications that influence how genes are expressed. These marks can change with stress, diet, temperature. Frozen cells may carry epigenetic patterns reflective of past conditions. Cloned or edited embryos may reset some of these marks during development, but not all outcomes are predictable. And there are ethical considerations. Is reviving genetic variants from long-dead individuals a restoration or a recreation? How do we ensure that edited animals integrate naturally into ecosystems? What limits should guide intervention? You do not rush past these questions. Instead, you imagine walking slowly between the nitrogen tanks, reading labels that bear species names and dates. Each vial represents a life once lived, a creature that moved, breathed, sensed the world. The cells are not conscious. They are not aware of their cold suspension. But they carry instructions written by evolution's long hand. There is tenderness in preserving them. In a world where habitats shrink and climates shift, frozen zoos are acts of hope. They say, we are not ready to let this variation disappear entirely. CRISPR adds another dimension to that hope. It suggests that preservation need not be passive. That sleeping genes might not only be thawed but gently repaired. That inherited weaknesses accumulating in small populations might be corrected before embryos are formed. Yet you remain aware that editing is not resurrection. It is not a return to some perfect past. It is a tool, one that must be guided by ecological understanding and humility. The future of conservation may involve laboratories as well as landscapes. It may involve careful blending of traditional methods, habitat protection, anti-poaching efforts, wildlife corridors, with advanced genetic strategies. You pause beside one tank and imagine a species teetering on the edge. Its living members are few. Their genomes are narrow. But within these frozen cells lies broader variation, preserved decades earlier. Scientists, working patiently, analyze sequences. They plan edits cautiously. They design breeding strategies that combine natural reproduction with assisted technologies. You begin with something familiar. Inheritance, in its simplest form, is balance. From your mother, you receive one copy of each gene. From your father, another. When cells form eggs or sperm, those gene copies separate. Only one version of each gene travels forward in each reproductive cell. When egg and sperm meet, the pair is restored. It is often described as a coin toss. If an organism carries one edited gene and one unedited version, each offspring has roughly a 50% chance of inheriting the edited copy. Over generations, that version may spread, remain stable, or fade depending on whether it offers an advantage. This slow arithmetic is the rhythm of classical genetics. Now imagine that balance shifting. A gene drive is a genetic system designed to increase the likelihood that a particular gene variant is inherited. Instead of a 50% chance, the edited gene might be passed on nearly 100% of the time. The coin no longer flips evenly. The concept builds directly on CRISPR. In a typical CRISPR edit, scientists introduce a change into a gene at a specific site. But with a gene drive, the CRISPR machinery itself is inserted into the genome alongside the desired trait. This means that when an organism carrying the gene drive mates with a wild counterpart, the CRISPR system can copy the edited sequence onto the unedited chromosome in the offspring. You picture it gently. An organism has two copies of a gene, one edited, one not. Inside its reproductive cells, the CRISPR system recognizes the unedited copy and cuts it. The cell repairs the cut using the edited version as a template, effectively converting the unedited copy into the edited one. Both copies now match. When that organism reproduces, nearly all of its offspring inherit the edited gene. And in their cells, the same process repeats. Generation after generation, the edit spreads through the population with remarkable speed. This is inheritance, but accelerated. You feel the power in that. Gene drives are being studied primarily in species that reproduce rapidly, such as mosquitoes. Some mosquito species carry parasites that cause malaria, a disease that affects millions of humans each year. Scientists have explored gene drives that either reduce mosquito fertility or make mosquitoes resistant to carrying the malaria parasite. In theory, releasing a small number of gene-drive mosquitoes into a population could transform that population over time. But conservation brings a different set of questions. Consider invasive species, organisms introduced into ecosystems where they have no natural predators. On islands especially, invasive rodents have devastated native bird populations by consuming eggs and chicks. Traditional control methods, such as trapping or poison, can be labor-intensive and sometimes harm non-target species. A gene drive designed to reduce fertility in invasive rodents could, in theory, suppress or eliminate the invasive population over generations. You pause with that idea. Unlike editing a few individuals in a breeding program, a gene drive is meant to move outward. To spread. To alter an entire population's genetic makeup. It is like releasing a current into a river and watching it flow downstream. There are different types of gene drives under development. Some aim to suppress populations by biasing inheritance toward traits that reduce fertility. Others aim to modify populations by spreading traits such as disease resistance without reducing numbers. The ecological implications are profound. Removing an invasive species might allow native plants and animals to recover. Altering a disease vector could reduce suffering. But ecosystems are intricate webs. Removing or modifying one species can ripple through food chains and interactions in ways that are difficult to predict fully. You sit with that uncertainty. Gene drives are powerful because they bypass the slow arithmetic of traditional inheritance. But that power requires extraordinary caution. Once released into the wild, a gene drive may be difficult or impossible to recall. Its effects could cross geographic boundaries if organisms migrate. Scientists are actively researching containment strategies. Some gene drives are designed to be self-limiting, spreading for a certain number of generations before fading. Others rely on ecological confinement, targeting species with restricted ranges, such as those found only on specific islands. Mathematical models play an essential role here. Researchers simulate how gene drives might spread under different conditions, varying population sizes, migration rates, mating patterns. These models help estimate speed, reach, and potential resistance. Because just as evolution can favor beneficial traits, it can also produce resistance to gene drives themselves. Mutations may arise that prevent the CRISPR system from cutting effectively. In such cases, the gene drive may slow or stall. This reminds you of something important. Even with advanced tools, biology remains dynamic. Living systems respond, adapt, resist. You breathe slowly and consider the ethical dimension. Altering inheritance is not the same as editing a handful of individuals. It is shaping the genetic trajectory of future generations. The organisms themselves do not choose. Humans design and release. There are ongoing discussions among scientists, ethicists, policymakers, and communities about governance. International guidelines emphasize transparency, public engagement, and ecological risk assessment. Indigenous and local communities, especially those living in areas where releases might occur, are increasingly recognized as essential voices in decision-making. You imagine standing on a small island where native birds once nested in abundance. Invasive rodents have reduced their numbers drastically. A gene drive offers hope of restoring balance. But the decision to use it must weigh potential benefits against unknowns. Will suppressing the rodents disrupt predator-prey dynamics in unforeseen ways? Could the gene drive spread beyond the island? How do you measure the value of risk versus recovery? These are not purely scientific questions. They are moral, ecological, social. And yet, the science itself is elegant in its design. A guide RNA. A cutting enzyme. A copying mechanism. A shift in inheritance probability. From 50% to nearly 100%. A subtle reprogramming of the rules. You return briefly to the image of a coin toss. For centuries, inheritance has felt like chance guided by selection. Gene drives tip the coin deliberately. They change the odds. This is not necessarily domination. In some contexts, it may be restoration, particularly where human activity introduced imbalances in the first place. But intention does not erase consequence. You feel both awe and gravity. Because altering one organism through CRISPR is like adjusting a single note. Introducing a gene drive is like changing the key of the entire symphony. As night settles in your imagination, you sense that gene drives represent one of the boldest applications of gene editing in conservation. They reach beyond individuals into populations, beyond the present into future generations. And so, the question deepens. Not just can we rewrite genes, or restore diversity, or awaken frozen variants. But should we guide inheritance itself? As you let that question drift gently through your thoughts, you realize that the next part of this journey must turn inward again, not to cells or chromosomes, but to conscience. Because when power expands, so too must reflection. And in the quiet forest of ethical thought, you begin to hear echoes that ask not only what is possible, but what is wise. You step away from a laboratory now. Away from nitrogen tanks and molecular scissors and inheritance currents. You walk instead into a forest at dusk. The air is cool. Light filters through leaves in soft, fading gold. The forest does not speak in equations or genetic sequences. It speaks in rustling branches, in insects humming, in distant wings cutting gently through air. And here, among living systems older than cities and nations, the ethical questions grow clearer. Should humans rewrite what evolution has shaped over millennia? It is not a simple question, and it does not demand a rushed answer. You let it settle slowly, like mist between trees. For most of history, human influence on nature has been unintentional or careless: forests cleared without understanding, species hunted without restraint, invasive organisms transported unknowingly across oceans. The imbalance we now attempt to repair is, in many cases, one we helped create. But awareness changes responsibility. Now you understand DNA as language. You understand CRISPR as a tool. You understand gene drives as currents in inheritance. With understanding comes choice. Ethics begins with pause. You consider the principle of precaution, the idea that when actions may cause irreversible harm, caution must guide decision-making. Gene editing in wild populations carries uncertainty. Even with modeling and laboratory testing, ecosystems are complex. Interactions ripple outward in ways difficult to predict fully. A single edited trait may influence survival, reproduction, competition. That shift may alter predator-prey dynamics. It may change plant growth patterns. It may reshape microbial communities. Nature is not a linear system. It is woven. So you ask, when is intervention justified? One answer emerges gently: when inaction carries greater harm. If a species faces extinction due to a human-introduced disease, and no other viable method exists to restore resilience, gene editing may represent a form of restitution rather than intrusion. If invasive species threaten entire ecosystems, and conventional methods fail or cause collateral damage, genetic tools may offer precision where blunt approaches cannot. But justification does not erase complexity. There is also the matter of consent, not from animals, who cannot deliberate in human terms, but from human communities. Conservation projects occur in real places where people live, work, and hold cultural relationships with land and wildlife. In recent years, scientists increasingly recognize the importance of public engagement. Gene editing proposals are discussed with local communities, policymakers, and indigenous groups whose knowledge and traditions are deeply tied to ecosystems. This inclusion is not merely procedural. It is ethical. Science does not exist in isolation. Decisions about releasing edited organisms into the wild affect shared environments. Transparency builds trust. Dialogue surfaces concerns that may not appear in laboratory risk assessments. You imagine a village near a wetland where mosquitoes carry disease. A gene drive proposal aims to reduce the mosquito population. The benefits could be significant: fewer infections, healthier children. Yet residents may worry about ecological consequences, about unforeseen changes. Listening becomes as important as editing. You reflect, too, on the concept of humility. For centuries, Western science often framed humanity as separate from nature observers, managers, controllers. But genetic knowledge reveals something different. You share the same alphabet as frogs and forests. The same 4 letters compose your body and theirs. Intervention, then, is not an external force acting on an alien system. It is one branch of life influencing another. That does not grant moral authority automatically. It deepens responsibility. Humility means acknowledging limits. It means designing safeguards. It means recognizing that not all problems demand technological solutions. Habitat preservation, pollution reduction, climate mitigation, these remain foundational. Gene editing is not a substitute for addressing root causes. International frameworks are evolving to guide these decisions. Environmental agreements consider biosafety, transboundary impacts, ecological monitoring. Research proposals undergo ethical review, ecological risk analysis, peer scrutiny. These processes may feel slow. But slowness, in this context, is care. You breathe in the forest air and consider time. Evolution unfolds over thousands, millions of years. Gene editing operates in seasons, sometimes months. That difference in scale can feel jarring. Yet urgency also presses in. Climate change accelerates shifts, biodiversity declines rapidly. Ethics balances urgency with deliberation. Too slow, and species vanish while debates continue. Too fast, and unintended harm may spread. There is no perfect equation. Instead, there is iterative learning. Pilot studies in contained environments. Stepwise releases with monitoring. Adaptive management, adjusting strategies as data emerge. You think of it like walking across a river on stepping stones. Each step is tested before weight fully shifts. Progress is possible, but never blind. Another ethical thread concerns equity. Who benefits from gene editing interventions? Who bears risks? If genetic tools are used primarily to protect ecosystems that serve wealthier nations while ignoring biodiversity in marginalized regions, imbalance persists. Global collaboration matters. Sharing knowledge, funding, and decision-making authority helps ensure that conservation genetics does not become another uneven resource. And what of intrinsic value? Some argue that species possess worth beyond human utility. That intervening genetically alters their evolutionary autonomy. Others counter that extinction caused by human activity is itself a violation of that autonomy, and that responsible intervention may honor intrinsic value by enabling survival. You sit between these perspectives, not choosing hastily. Ethics is not about eliminating uncertainty. It is about navigating it with care. In the forest, you notice how many species interact without awareness of genetics or policy. Roots exchange nutrients through fungal networks. Insects pollinate flowers without considering probability. Life proceeds through interdependence. Perhaps ethical conservation seeks to preserve that interdependence with minimal distortion. CRISPR, gene drives, frozen zoos, these are tools. They reflect human ingenuity. But tools must align with values. The deeper question is not merely what can be edited, but why. Is the goal ecological restoration? Disease reduction? Prevention of suffering? Protection of biodiversity for its own sake? Clarity of purpose guides boundaries. You feel the weight of that clarity. Because without it, technology can drift toward novelty or control. With it, technology can serve stewardship. As dusk deepens in your imagined forest, the sounds soften. You sense that ethics is not a barrier to progress, but its companion. It slows, questions, refines. It asks whether intervention respects complexity, whether it includes voices beyond laboratories, whether it acknowledges uncertainty openly. And as you let these reflections settle, another layer of wisdom begins to surface, knowledge rooted not in sequencing machines, but in generations of lived relationship with land and species. Voices that have practiced conservation long before CRISPR was discovered. Voices that may help guide how genetic futures are shaped. And in that quiet forest, you begin to listen for them. You leave the glow of fluorescent lights and the hum of laboratory equipment behind. Now, you are walking along an ancient trail, worn smooth by generations of feet. The air carries scents of soil, herbs, and rain-soaked leaves. Birds sing faintly, and somewhere in the distance, a stream whispers over stones. Here, the guardians of the land move with deep familiarity. Their eyes track subtle signs, broken branches that indicate a passing deer, a flowering plant whose bloom signals seasonal change, the call of a frog hidden beneath wet leaves. These are indigenous communities, stewards of landscapes shaped through centuries of observation, care, and ritual. Their knowledge is not written in papers or stored in databases, it is alive in practice, memory, and relationships. You notice how every plant, animal, and insect is understood within context. Not as a mere species name or gene sequence, but as a participant in a web of interconnections. When a tree is pruned, it is done to encourage balance in the forest. When fish are harvested, timing respects reproduction cycles. When fire is set, it is controlled and purposeful, sustaining diversity rather than destroying it. You realize that conservation is not only about genes. It is relational. It is cultural. It is ancestral. Modern genetic tools, like CRISPR, operate in a laboratory. They edit DNA with precision, offering potential for disease resistance, restoration, and revival. But these tools are not the first means humans have used to influence life. Indigenous knowledge represents countless generations of feedback loops between people and ecosystems, a form of adaptive management as nuanced and sophisticated as any genome editing system. You reflect on this, science often frames intervention as a technical challenge, a puzzle of letters, enzymes, and probabilities. Indigenous stewardship frames it as a relationship, a story of reciprocity, respect, and observation over time. Both perspectives aim to sustain life, but their methods, ethics, and timescales differ. Consider a species at risk, a frog whose population has declined drastically. Geneticists may examine its DNA to identify vulnerabilities and explore gene editing strategies. Meanwhile, the local community may have observed shifts in the frog's habitat, altered water flows, or the arrival of new predators. Their knowledge might suggest solutions that do not require direct genetic manipulation, restoring wetlands, timing harvests, protecting breeding sites. Science becomes more effective when it listens. CRISPR could complement, not replace, these practices. It could help where natural resilience is insufficient, or where rapid change threatens extinction. but its use must be informed by the deep ecological literacy embedded in indigenous knowledge. Genetic edits cannot substitute for holistic understanding of habitat, climate, interspecies relationships, and the cultural dimensions of stewardship. You picture a dialogue unfolding. Scientists, conservationists, and indigenous communities come together, sharing data and stories. Sequencing results are presented alongside oral histories and observational records. Maps of genetic diversity overlay maps of sacred groves and ancestral lands. Decisions about interventions are made collectively, blending laboratory insight with lived experience. This integration brings humility. You sense that no single approach is sufficient. CRISPR can guide traits in cells, but it cannot teach patience, respect, or relational awareness. Only human communities can cultivate that broader understanding. There is also a temporal lesson here. Indigenous knowledge embodies long-term thinking, managing species and landscapes over decades and centuries, not just seasons or grand cycles. In contrast, the pace of modern genetic technology is rapid. Experiments occur in months. Gene drives could spread in a few generations. Bridging these timescales requires care. Decisions that seem urgent in the lab must be considered in the context of centuries of ecological practice. You begin to see CRISPR not as a command over life, but as a conversation partner. It offers suggestions at the molecular level, highlighting possibilities, but it cannot understand ecosystems or cultures. The wisdom of generations teaches when, where, and whether those suggestions are appropriate. You wander further into the forest, imagining an old tree that has stood for hundreds of years. Its roots entwine with those of other trees. Its seeds carry genetic memories of droughts, floods, and changing climates. Indigenous communities have nurtured its growth, protected it from fire and overharvest, and observed its cycles carefully. Scientists might one day sequence its genome to understand resilience. But sequencing alone cannot replicate the subtle care that allowed it to endure. Similarly, when thinking about endangered species and gene editing, you recognize that technology is only part of a larger story. To conserve life effectively, we must respect ancestral knowledge, cultural practices, and ecological relationships. CRISPR can offer solutions at the microscopic level, but the forest, the river, the air, and the people have their own guidance. You imagine workshops where children learn both DNA sequences and traditional stories, where elders teach patterns of seasonal change alongside modern ecological monitoring. Knowledge flows in both directions, forming a richer tapestry than either approach alone could create. This vision reminds you that conservation is relational, not hierarchical. It is not about humans commanding nature. It is about humans listening, learning, and acting in collaboration with other species, with landscapes, and with cultural stewards. As you walk, you notice small indicators of life persisting quietly, a frog's call beneath a fallen log, the shimmer of insect wings, the bending of reeds in a stream. Each organism carries its own genetic story. Each landscape holds accumulated wisdom of countless interactions. Intervention, when necessary, must honor both. CRISPR may awaken sleeping genes, repair vulnerabilities, and help species endure rapid change. But its application becomes most meaningful when it is guided by understanding that transcends the laboratory, by ecological knowledge, by relational awareness, and by respect for the cultures that have protected biodiversity for centuries. You pause beside the stream, listening to the water ripple over stones. You sense that conservation is not a singular act. It is a dialogue across time, between generations of humans, between species, between genes, and between communities who know the land intimately. Science becomes less about authority and more about conversation. Gene editing does not dictate. It suggests. And human wisdom, ancient, relational, observant, decides whether to follow, adjust, or refrain. And in the quiet of that reflection, you realize that the next chapter will return to the rhythms of evolution itself, examining how the slow pace of adaptation meets the urgency of a changing world, and how human intervention might navigate the tension between patience and immediacy. You find yourself standing on a quiet hilltop, overlooking a valley carved over millennia. The mountains rise like silent sentinels. Rivers snake through the lowlands, winding slowly, patiently, shaping the land with relentless yet gentle force. Each contour of stone, each layer of sediment, tells a story written in eons. This is the pace of evolution. It is slow, deliberate, and unhurried. Species emerge, adapt, and shift over countless generations. Alleles drift in populations. Mutations arise by chance. Natural selection favors those best suited to current conditions. The clock of evolution ticks over centuries, millennia, even longer. And yet, today, you notice something jarring. Climate change moves faster. The temperature rises not over eons, but over decades. Ice sheets retreat in years. Storms strike with intensity previously unseen. Habitats transform within a single human lifetime. Species that once had ample time to adapt are now confronted with rapid environmental upheaval. You imagine a slow-moving tree, its seeds dispersed over gentle breezes. Once, generations of seedlings sprouted, grew, and reproduced under stable conditions. Now, shifts in rainfall patterns threaten its survival. Seeds may fall in places no longer hospitable. The pace of environmental change outstrips the pace of adaptation. This is where urgency enters the story. Evolution can no longer be relied upon to protect every species. For those already small, fragmented, or threatened by human activity, the traditional mechanisms of natural selection may not operate quickly enough to ensure survival. The slow rhythms of gene flow, mutation, and selection are overtaken by rapid climate shifts, habitat destruction, and disease emergence. You feel the tension deeply. On one hand, patience has always been the ally of life. Evolutionary time produces resilience. Populations adjust. Ecosystems balance. Complexity emerges. On the other hand, waiting may mean watching species disappear, lost before their adaptive potential has time to manifest. It is a delicate, almost impossible balance. And here, genetic intervention presents itself as a possibility, not as a replacement for evolution, but as a form of assistance. You imagine scientists examining a population of corals. Rising ocean temperatures have caused widespread bleaching. Corals that survive may carry genetic variants allowing them to tolerate higher temperatures. Through selective breeding or gene editing, these variants could be increased in frequency. CRISPR could, in theory, introduce protective alleles from heat-tolerant strains into vulnerable colonies. In essence, humans may provide species with the gift of time. they may accelerate adaptation where nature cannot keep pace. Yet you sense the complexity. Adaptation is never a single gene or a simple trait. It involves networks of genes interacting with one another and with the environment. Editing one gene may provide some resilience, but it may not address the full suite of challenges a species faces. Moreover, ecological interactions are intricate. A change in one species can cascade through predators, prey, and symbionts, producing unforeseen consequences. You think of evolution as a river flowing slowly, carrying all organisms along in a shared current. Climate change is a sudden surge, a flash flood. Gene editing is like placing temporary channels, guiding the water, allowing some organisms to ride the surge rather than be swept away. But those channels must be planned carefully, or the flood may spill unpredictably, damaging what was already stable. There is also the human element to consider. Urgency can push action without full understanding. Ethical reflection, ecological monitoring, and stakeholder engagement must accompany intervention. Acting too quickly may risk harm, acting too slowly may allow extinction. Both paths carry weight. You watch clouds drift across the valley. Each carries rain. Each influences the landscape differently. In your mind, each species is like a cloud in its own way, shaped by genetics, environment, and chance. Some drift slowly, resilient to minor shifts. Others are fragile, easily redirected by storms. And now, the storms are arriving faster than ever. Gene editing can bolster some clouds, providing them resilience to change. But it cannot conjure entirely new species, nor can it recreate lost ecological relationships. It is an aid, a buffer, a measured nudge, not a panacea. You consider the choices facing conservationists. Which species merit intervention? Which traits should be targeted? How do we weigh urgency against patience, risk against hope? Every decision is a negotiation between understanding and uncertainty, between what is possible and what is prudent. Even as you ponder, the forest below hums with life. Birds call. Insects stir. Plants shift in the breeze. Life is persistent, adaptive, and fragile all at once. It is this duality that defines the present moment. Evolution has shaped remarkable resilience over millennia. Human-driven change now demands swift action to preserve that resilience. The challenge is to intervene without overstepping, to assist without overriding, to act without arrogance. And yet, hope persists. By combining careful observation, ecological knowledge, and precise genetic tools, humans may help species navigate the narrowing window between vulnerability and adaptation. By augmenting natural variation, repairing harmful mutations, and selectively increasing resilience, we may give species a fighting chance in an era of accelerated change. You feel a quiet tension between scales of time. The slow pulse of evolution, measured in centuries, stands beside the rapid beat of climate, disease, and habitat transformation. Your mind reaches for equilibrium, seeking ways that these rhythms might harmonize. Gene editing is a bridge, not a replacement, but a hand extended across the gap. You inhale the valley air deeply, letting it fill your lungs and thoughts. There is no certainty, no guarantee. But there is deliberate care, guided by understanding and respect for life's complexity. And as the sun dips lower, casting long shadows across the hills, You sense that this chapter of the journey between the pace of evolution and the urgency of now will lead naturally into thoughts of stewardship itself. Because the ultimate question is not only whether species can adapt, but how humans will choose to guide that adaptation with wisdom, humility, and patience. And in that quiet, reflective space, you begin to imagine the next step, a future written with care, where technology, ethics, and compassion converge. You close your eyes and return once more to the microscopic world inside a single cell. It is familiar now, the double helix winding gently, letters A, T, C, G repeating like a quiet song. Each sequence a sentence in the story of life, each gene a chapter written over millions of years. You sense the enormity of this language, the intricate poetry in its structure, and the care evolution has taken in composing it. Tonight, you are not rushing to edit, to correct, or to fix. You are imagining the possibilities, the responsibility, the stewardship that comes with understanding the code of life. CRISPR is here in your mind, not as a sharp tool or a weapon, but as a pen. A pen that allows humans to add annotations, gently, thoughtfully, without rewriting entire stories recklessly. A pen that, when held with respect, can restore resilience, mend fragile threads, and offer life the chance to endure challenges we could not prevent. You think of the species you have encountered in your journey, the frogs silenced by disease, the corals stressed by warming oceans, the seeds preserved in frozen zoos. Each carries lessons of survival, fragility, and adaptation. And now, humanity has the potential to assist, not dominate, guiding these lessons forward into new generations. Yet stewardship is delicate. It is not control for the sake of control, nor is it intervention without reflection. It is careful attention, humility, and respect. It recognizes that life is more than the sum of its parts. That genes, while powerful, do not act alone. That ecosystems, cultures, and communities intertwine, shaping outcomes in ways far more complex than any laboratory can fully predict. You feel the rhythm of this responsibility, slow and steady, like a heartbeat echoing across time. It is the same rhythm that guided indigenous stewards, that shaped evolution, that has allowed life to persist through ice ages, droughts, and storms. It is patience tempered with awareness, action guided by empathy. You imagine a scenario far in the future. In a vast wetland, endangered birds that once teetered on the edge now soar. Gene editing helped restore genetic variants lost through population decline. Habitat restoration created safe breeding grounds. Local communities, carrying traditions of care, monitor, guide, and celebrate the lives that flourish. Here, technology and culture coexist, supporting life without replacing it. You picture coral reefs, fragile and vibrant. CRISPR has assisted in enhancing heat tolerance. But it was not a miracle. It was a careful application of knowledge, combined with reductions in pollution, protection from overfishing, and engagement with the people who depend on the reef. Life continues to teach humans as much as humans guide life. The frozen zoos, once quiet and still, now serve as both archive and library. They hold the past's whispers, the potential for renewal, and the knowledge that careful hands may awaken sleeping genes when the time is right. You imagine embryos thawed with reverence, nurtured into life in ways that honor their species' history and ecological role. Gene drives, once a concept that rippled like a hidden current, are applied sparingly, tested, reversible where possible, and designed with humility. They are not instruments of conquest, but tools for balance, nudging populations gently, ensuring survival while respecting the integrity of ecosystems. In every case, you notice a common theme: intention matters. It is not enough to possess the ability to edit genes. What matters is the wisdom with which that ability is wielded. Each intervention must answer the quiet question: does this serve life as it exists naturally, sustainably, and ethically? You return to the language of DNA. The letters are not just instructions. They are narratives of survival. They are poetry etched into cells. Editing them is like adding a stanza to an epic. It requires listening, reflection, and a deep understanding of context. And as you breathe slowly, the enormity of life feels intimate. You sense the connection between your own cells and the diversity of life across the planet. A bird's flight, a tree's roots, a frog's call all echo the same patterns, the same alphabet. Stewardship becomes not an abstract ideal, but a tangible relationship with the world. You imagine children in distant lands learning this truth. They hold microscopes alongside stories passed down through generations. They witness CRISPR's power, but also its responsibility. They see the balance between intervention and patience. They grow up understanding that conservation is not about domination, but dialogue between science, ethics, and the living world. Sleep begins to press gently on your mind as you visualize the continuity of life. Each species, each genome, each ecosystem is a thread in a tapestry that stretches backward into the past and forward into the future. Some threads are fragile, others resilient. Some have frayed, others remain strong. CRISPR and conservation together offer a way to mend, reinforce, and honor this tapestry. And yet, even in the calm of this imagining, you understand there are no certainties. The future of life cannot be predicted with absolute precision. Genes may behave unexpectedly. Ecosystems may shift in unforeseen ways. Climate may challenge even the best-laid plans. The work of stewardship is ongoing, iterative, and, above all, respectful of the limits of human understanding. You picture the quiet satisfaction of a species that persists because of careful, informed action. You see the possibility of ecosystems thriving where once they faltered. You feel the gentle presence of scientists, indigenous knowledge holders, communities, and caretakers working together. Each action, small or large, ripples through time like a gentle wave, influencing generations yet to come. And as you sink deeper into rest, the four letters of life A, T, C, G continue to spiral quietly in your imagination. You sense the infinite potential in their arrangements, the delicate balance between chance and design, intervention and patience, urgency and humility. CRISPR is a pen. Conservation is stewardship. Life continues to write itself in cells, in forests, in oceans, and in hearts that choose to protect it. And as you drift towards sleep, you realize that the final thought is not whether humans can edit DNA, but how gently, thoughtfully, and lovingly we will choose to do so.